Let's review target amplification methods. The most widely used method is the polymerase chain reaction, or PCR. This technique relies on specific primers to amplify DNA sequences. There are several variations. RT-PCR, or reverse transcriptase PCR, which begins with RNA and converts it into complementary DNA before amplification. Nested PCR, which involves two sequential steps using two sets of primers for increased specificity. Multiplex PCR, which allows more than one set of primers to be used in the same reaction, enabling the detection of multiple targets simultaneously. Another important variant is real-time or quantitative PCR, also known as qPCR. This is essentially standard PCR, but with an added probe or dye. The amplified product generates a fluorescent signal. By detecting this signal in real time, the system can quantify the starting material. This process requires specialized thermal cyclers equipped with fluorescent detection systems. Beyond PCR, there are additional target amplification methods. These include NASBA, or nucleic acid sequence-based amplification, and TMA, or transcription-mediated amplification. Both NASBA and TMA, like RT-PCR, begin with RNA as the starting material. The RNA is first transcribed into complementary DNA, which is then amplified. In summary, PCR and its many variants, as well as NASBA and TMA, are powerful methods for amplifying nucleic acids, allowing for sensitive detection and quantification in molecular diagnostics. Let's review the PCR standard reaction mix, steps, and controls. The PCR reaction mix requires several key components: a DNA template, which may be genomic DNA or complementary DNA; oligonucleotide primers, short sequences that guide where amplification begins; a thermostable DNA polymerase, the enzyme that builds new DNA strands; nucleotides, or DNTPs, which include DATP, DCTP, DGTP, and DTTP; a PCR reaction buffer, which ensures optimal conditions for enzyme activity. To prevent contamination, the PCR laboratory setup is very important. A unidirectional workflow should be maintained, moving only from clean to contaminated areas. Pre- and post-PCR areas must be separated. Reagent preparation should be in a positive pressure environment. Sample preparation should occur in negative pressure. Amplification and detection should also take place under negative pressure. PCR hoods equipped with UV decontamination are used to reduce contamination risk. Now, the steps of PCR. 1. Denaturation, at around 90 to 95 degrees Celsius, separates double-stranded DNA into single strands. 2. Annealing, at about 50 to 60 degrees Celsius, allows primers to hybridize to the single-stranded template. The annealing temperature depends on the G and C content of the primers. High stringency prevents mismatches, while lower stringency allows for some mismatching. This step controls the specificity of binding. 3. Extension, at 70 to 75 degrees Celsius, is where the polymerase synthesizes the new DNA strand. Each cycle of these steps results in a doubling of the DNA present. Finally, controls for PCR are essential. The blank reaction contains all reagents except DNA template. This ensures that no contamination has entered the system. The negative control contains all reagents and a DNA template that does not have the target sequence. This confirms the specificity of the reaction. The positive control includes all reagents plus a DNA template with a known target sequence. This ensures the sensitivity of the PCR, confirming that the reaction works as expected. Together, these components, steps, and controls ensure that PCR produces accurate and reliable results. Let's review probe amplification methods and signal amplification methods. Starting with probe amplification methods. The first is the ligase chain reaction, or LCR. This is an isothermal method, meaning it runs at a constant temperature. It works by ligating or joining together two adjacent oligonucleotide primers. These primers hybridize to one strand of the target DNA, allowing for amplification. The second method is strand displacement amplification, or SDA. Like LCR, it is also isothermal. In this method, the process creates an altered target that includes a restriction endonuclease recognition site, which facilitates amplification. The third method is qB replicase. This also operates under isothermal conditions. It uses an RNA-dependent RNA polymerase derived from the bacteriophage qB, enabling efficient amplification of RNA sequences. Now let's consider signal amplification methods. Unlike probe amplification, these techniques do not amplify the target itself, but instead amplify the signal that indicates its presence. The first method is hybrid capture. This uses an antibody that recognizes DNA-RNA hybrids. The process is conducted on a solid phase and employs chemiluminescence to generate a measurable signal. The second method is branched chain DNA, or bDNA. This relies on a series of hybrid probes that bind to the target sequence and build a branched structure. This structure enhances the signal, also detected through chemiluminescence, allowing for the detection of specific RNA sequences. In summary, probe amplification methods like LCR, SDA, and qB replicase increase the number of probe-target hybrids. Signal amplification methods like hybrid capture and bDNA amplify the detectable signal, not the target, enabling sensitive molecular detection.